Welcome back to our series on unlocking the mystery of genetic testing. I've talked a lot about the process of genetic testing, of gathering a sample, extracting the DNA, amplifying the DNA, conducting the genetic sequencing, creating reports. But one thing that I haven't actually shown anybody is what goes into genetic testing? Where do the reagents come from, the chemicals that actually make it possible to turn a sample into something that you could actually genetically sequence? We talked about the process, but what actually is it that makes it happen? This may be surprising to you, but the reagents for the entire process, whether you're doing genetic testing for COVID, for embryo screening, cancer diagnosis, whatever it may be, are actually able to fit in very small packages. This right here actually contains everything that you would need to prepare samples for genetic sequencing of an embryo sample, believe it or not. These are all of the individual reagents. They're the reagents for extraction, for amplification, and for library preparation. They are normalization buffers. They are the magnetic beads that get applied to the sample. Everything that you would need right here to prepare a DNA sample for genetic sequencing. Now, of course, when you load it onto the sequencer, there's a device called a flow cell or a chip that the samples are put on. But for the entire front-end process, this is it. Can you believe how simple it is? If you would like to learn more about the type of genetic sequencing and the type of services that can be provided by genetic sequencing, check out avriogenetics.com. Thank you.